I have to take, actually, thanks. I'm really glad to be here. I have to take issue with one thing that Alan said. Schrodinger, I think it can be shown, borrowed, if that's the right word, almost all of his arguments from a 1921 paper by Muller. And if you go back to the American Scientist, you discover that Muller said that the hereditary material must be crystalline because it had to be self-assembly. It had to be self-assembling. It had to be stable but mutable. And then at the end he says, maybe the way we will learn about this is not through the genetics that we know, but through studying Darrell bodies. Now, Darrell bodies were bacteriophage, which were discovered a few years before. And so he ends his essay by saying, must we become microbiologists and physicists in order to understand the hereditary material? If you haven't read this article, you really should. But of course, Schrodinger was very famous and recruited lots of his physics friends to join after he wrote his book. Well, okay, I'm going to talk about 25 years of coevolution with Bernard Dujon, who used to look like this. But then on the other hand, I used to look like this. So I've known Bernard for a long time, as I will show you, and at a number of times we have intersected in our scientific interests. But I'm going to start with a more general introduction and just start with the karyotype. So if you happen to be female and you were to pull out almost any cell from your body and do this karyotypic staining, you would see that you had 23 pairs of chromosomes, and virtually every cell in your body looks like that after 1 trillion cell divisions. Okay, cancer cells don't. And so they have all of these amazing kinds of rearrangements, including this most recently discovered chromothripsis, chromosome shattering, which we didn't even know about until 2011. And the question is, why has our genome remained this stable? And the answer is that our genome remains stable because it has accurate repair mechanisms to repair chromosome breaks. And cancer cells have very active mechanisms to repair chromosome breaks. They're just not accurate. Okay, they do end joining instead of homologous recombination, and that's why our genome doesn't look like this every time our cells go through division. And the fact is that every time a mammalian cell goes through replication, it acquires something like 12 chromatid breaks. That means that, so these are done in the absence of Rad51, where these breaks are not being repaired. And so that means that during the replication cycle, there's one of the two sister chromatids has been broken, and it's the job of Rad51 protein and lots of other proteins to use the sister chromatid as a template to repair the broken chromosome. And yeast cells are alive when you knock out Rad51 for only one reason, and that is that the yeast genome is this big and our genome is this big. Okay? The rate of breakage in yeast cells is actually the same as the rate of breakage in mammalian cells, and it's only the small size that allows it to elude the lethality of knocking out Rad51. Okay, so there are lots of ways of repairing a double-strand break. There are end joining mechanisms that I mentioned, and also the ability to just simply add new telomeres to the end. But I'm going to talk about homologous recombination mechanisms. The basic principle of these mechanisms is that the broken end of the DNA has to find a template, which it is going to find by being able to make Watson-Crick base pairs with the template, and to use that then to copy missing information from the break to seal the break up by a recombination mechanism. Quite often, just a tiny little patch of DNA is made. Sometimes that's associated with a loss of heterozygosity. But also, there's a second mechanism that I'm going to mention called break-induced replication, where only one of the two ends succeeds in finding the homologous donor template and then can replicate hundreds of kilobases to the end of a chromosome. And it turns out that these are both very important repair mechanisms. So just to sort of give us a frame of reference, When the break is made, the first thing that happens is that there are enzymes that chew away one of the strands, leaving a long single-stranded piece of DNA. This is the substrate for the RAD51 protein, as I'll show you in a minute. Then the RAD51 nucleoprotein filament searches around the genome, which is a remarkable thing. It can basically explore most of the genome to find a partner, make those Watson-Crick base pairs, and initiate new DNA synthesis. But what makes this special is that this kind of DNA replication is not like what Messelson and Stahl showed us for normal replication, which would be that a new strand would remain base paired to its template strand. In fact, the newly synthesized DNA is somehow extruded, much more like the way RNA is copied from DNA. And then eventually, all the newly synthesized DNA is found in the recipient locus, as we showed by doing a Messelson and Stahl type experiment, showing that all the heavy isotope ended up in one place. This other mechanism, break-induced replication, also looks like it ought to be more like a Watson-Crick, because it does, in fact, synthesize both strands newly. But it turns out from recent work that was done by a former postdoc of my lab, Anna Malkova, that the synthesis is not coordinated. One strand is made first, and then the second strand is made again. It's not a normal replication process. And as a consequence, both of these processes are highly mutagenic, and I'll come back to that. Okay, so I got started in this business before I had met Bernard, studying mating type switching in Saccharomyces cerevisiae, a sex change operation that this organism has evolved so that it could change from one mating type to the other. And what we now know is that the mating type is specified by a particular locus, and the cells can either be A or alpha. These transcripts encode regulatory proteins that then give the cell its sexual identity. But there was this ability for these sequences, which are entirely different from these sequences, to be replaced. And that turned out to be caused by a site-specific nuclease that makes a double-strand break at this location, results in the removal of these red sequences, and then a copy is made, not from a sister chromatid in this case, but from an ectopically located donor sequence. And there's lots of other features of this system. The donors, of which there are two, are heterochromatic. They are maintained in a silent configuration by some cis-actin sequences and by the SIR2 histone deacetylase. And that keeps highly positioned nucleosomes across this region. It doesn't prevent these from being good donors, but it prevents them from being recipients, because the sequence here is identical to here, but it doesn't get cut because there's a nucleosome sitting right there that prevents its cleavage. So this is a directional system. A cut is made here, and then these sequences are copied in to repair the break. And so that seemed to me a long time ago to be a very good model to study DNA repair in a controlled situation. And then Ira Herskowitz's lab tricked up the HO endonuclease gene by putting it under the control of an inducible promoter, which meant we could turn on the HO endonuclease in all the cells of the population at the same time, and we could follow this process in real time. And so in 1988, we could show that when you turned on this HO endonuclease, the restriction fragment here in the mat locus got cut, then things happened to it, it went away, and what would appear but the new sequences. And since the restriction sites here are different from here, we could follow this in time. So over time, we've done this a lot. We've gotten better at how fast we can do the whole system. And so we've learned lots of things about the individual steps in this process. I already sort of mentioned the key steps that I'm going to feature. The idea that there's a break which is chewed away by a series of exonucleases, the assembly of a RAD51 filament. Each RAD51 molecule binds 3 base pairs. And so you get a long filament that then has this capacity to search the genome and then form those base pairs. It's still a somewhat magical reaction. In the last couple of years, actually in the last year, I would say, work from Eric Green and Patrick Sung have made a big progress in understanding how this works, and especially from a crystal structure that was done by Nicolo Pavletich. So we have some real insight as to what's going on here. The most interesting feature is that the Rad51 molecule stretches the DNA by about 50%. But if you look at how the stretching occurs, the 3 base pairs that are bound are still in a B-form configuration, then there's a huge stretch, and then there's 3 more B-form DNA. So the searching is being done in units of 3, and we've learned a lot about that. Okay, so at the same time, Bernard was studying the omega intron in mitochondria, which I am not going to go through the biology of. He may do this tomorrow, but I'm not. But he in the sequencing realized that the intron contained an open reading frame, and the open reading frame appeared to contain the key factor for the movement of this intron. And then, in order to study this, they did one of the first real synthetic gene experiments, because in order to express ISCE1, in this case in bacteria, but also in the nucleus of yeast, all the UGA codons, which in mitochondria encode tryptophan, had to be changed to UGG in order to encode tryptophan, and not a stop codon, which is what they encode for the nuclear genetic code. So Bernard's lab developed this endonuclease, and then I went on a kind of partial sabbatical to Bernard's lab and hooked up with Anne Plessis, who was a graduate student, and Arnaud Perrin, and we published a paper showing essentially what I just showed you for mating type switching also works for the ISCE1 nuclease, namely that if you put it under the control of a galactose promoter, you could get a double-strand break. The break would then lead to the repair of the break by using a homologous sequence as a donor. And so as it says here, the results demonstrate that mitochondrial encoded ISCE1 nuclease, when expressed in the cytoplasm from a nuclear expression vector, is active on a nuclear target. So that was a big deal. Okay. Then if we fast forward, Bernard figures out that you can do this in mammalian cells. Okay. So you go to PubMed and you find 2,191 papers which have used ISCE1 in mammalian cells to study recombination. Lots and lots of people contributing to this understanding, and basically being able now to say that things that we first learned in yeast largely appear to go by the same mechanisms in mammalian cells. And obviously there are different bells and whistles. Yeast cells don't have BRCA1 or BRCA2. They have other genes that do somewhat similar things. They don't have DNA PK for doing end joining, but they still have all the other end joining components. But nevertheless, underneath it all, it's basically the same mechanism. And we've learned a huge amount about that by being able to make controlled double-strand breaks in mammalian cells using this system. What the system lacks so far is efficiency and synchrony, the way that we can do it in yeast. So if you turn on ISCE1 in mammalian cells, the events dribble on for 24 hours, which, by the way, is also true for the Cas9 system, if you were interested. So things are not yet to the yeast level, but they're progressing. Okay, so now I'm going to tell you 2 little stories about yeast work that I hope you will find interesting. The first is, how does that search for homology really take place? This is done primarily by a graduate student, Cheng-Sheng Li, a little bit by another graduate student, Anuja Mehta, and a very large amount also by an undergraduate, Wendy Wong, who is astonishing. Okay, so sometimes I say, I wonder what would happen if we did such and such, and three weeks later she says, oh, I made those strains and here's what happens. So lucky me. Okay, so we can measure early events. In 1990, I had a postdoc named Charles White, now known as Charles White, and despite being an Australian, he is the president of the French Genetic Society. He figured out that if you wanted to know the earliest event of new DNA synthesis in this mating type switching system, you could use a PCR primer that was specific for the donor and another PCR primer that was specific for just outside the mating type locus. And only after you made 50 base pairs of new DNA synthesis would there be a covalent template to do a PCR reaction. So you could get not the end of the reaction, but the first inklings of when did new DNA synthesis start. And so that's what's measured here. And what we learned in many experiments of this sort is that if the donor is intrachromosomal, the events are faster than if they're interchromosomal. And every chemistry course you ever took as a freshman in college, somebody said an intramolecular reaction is faster than a bimolecular reaction, and a whole chromosome is behaving the way we learned for basic chemistry. So a whole chromosome still has an advantage. Sites 500 kb away on the same chromosome react faster than if they are on different chromosomes. So we wanted to explore this question in more detail, and a number of years ago now, Nobel's lab in Seattle published this reconstruction of the architecture of the yeast chromosomes. And the idea was to take what is known as chromosome conformation capture, to cross-link rare events that were just captured by adding formaldehyde for a short time to the cells, and then by a series of tricks end up being able to sequence all these junctions, and from these junctions be able to deduce which sequences were more frequently in contact with each other than other sequences. And so you get contact maps. Some things are very interesting. Sequences near centromeres show lots and lots of interaction. And that's because all the centromeres are clustered. Okay? But there are other places where there are lots of interactions which are different from other regions. And the same is true within a chromosome. And so at the end of this, they came up with a 3D picture. It's obviously a reconstruction. It's not, they take the contact data and then they sort of make what they think is the best fit map for those data. And our question is, if we make the break in the blue chromosome, does it matter whether a donor to repair that break is on a red chromosome or an orange chromosome or a yellow chromosome? Does, in fact, this picture have any reality in terms of the repair efficiency? Okay, so the experiment is simple. We make a break here. We've moved the HO cleavage site into just a garden variety biosynthetic gene, LU2. We put another copy of LU2 elsewhere, and it could be at many different locations, and then we just measure viability. How often can it repair the break? And so I'm just going to tell you what we found, and then I'll show you a little data. We found that interchromosomal repair is in fact faster than intrachromosomal repair, that mutations that affect double-strand break-induced mobility don't actually affect how these, I'm not going to really talk about this, I should have taken it out. And the real event is that intrachromosomal repair reflects the contact probability and also the orientation of these with all the centromeres clustered. So here's the experiment. We make 20 strains. We made a break at one place. We have 20 strains with donors at different locations. We measure their repair efficiency. Okay? It turns out that, roughly speaking, those sites which would be predicted to be far away have considerably lower efficiencies of repair than those sites which are predicted to be closer. And in fact, if we do this as a Pearson correlation coefficient, we get a remarkably good line. So this suggests that chromosome position, as measured by this metric of contact probability, really has some predictive value, that you can say that sequences that are closer together in the nucleus are more likely to recombine. This is very similar to what mammalian people, Tom Mastelli, Fred Alt, Job Decker, have shown with respect to translocations in mammalian cells, that proximity is also a big factor in how often pairs of double-strand breaks are joined together in translocations. But here we're looking at homologous recombination mechanisms and not translocations. We are now trying to do the translocation experiment in yeast, but it isn't done yet.
Are these all haploid?
These are all haploid, yeah. Okay, so then, as it says here, anticipating reviewer number 3, we decided we had to move the break and do the experiment over. We didn't do as many sites, but we did a bunch. And so here's what's interesting. If you look at this site, it's very efficient. When it was recombining with this site, it wasn't very efficient. And that means that the recombinational ability of this site to act as a donor is not its local environment, it's its proximity to the site of the double-strand break. And so that, I think, is a very important concept, that what we are looking here is not just that this site is heterochromatic and can't do anything, it's really location, location, location. Okay. Okay. Good correlation again. Okay, and then I'm just going to show you a little data. This is Wendy's project. Wendy did all of this by herself, the undergraduate. And so we've also looked at what happens if you make a break and put donors along this chromosome. And you get, again, a very good correlation. I think I mentioned this to Bernard last night. We have two outliers, and they are the sites closest to the two ends of the chromosome, which I think means that their contact frequency is underrepresented relative to their proximity, though I don't know why. In any case, if you also now measure this as a function of where these sites are in the chromosome, what we see is something that we didn't know that we would see, and that is that we know that yeast cells have, this is chromosome 2 as measured by Noble in that map that I showed you at the beginning. This is the architecture that they imagined for chromosome 2. And what we see is that sites on this side of the centromere all sort of linearly get better and better with distance, but they also get better and better with distance on the other side. And that probably means that that chromosome arm can move from one side to the other. And so we get exactly the ratio we would expect as if that was on this line rather than something far away. Now, the audacious experiment that we are trying now is to get rid of the centromere. At least temporarily, we have ways to do this. And then I think what will happen is that we'll get a line that looks like that. We'll see. Anyway, it's a cute experiment. Okay, the second little story I want to tell you has to do with how this repair process really takes place. And that is that although it's much more advantageous to do this gene conversion kind of repair as opposed to doing end joining, which is intrinsically mutagenic, accurate repair turns out to have risks. There's about a 1,000-fold increase in the rate of mutations associated with these regions that are undergoing repair. Okay, so what we found, this started with work done by Wade Hicks, who was a graduate student, Min Lee Kim, who was another undergraduate, and me. And what we found was that the gene conversion rate associated with the repair was 1,000 times higher than the spontaneous rate of mutation, that many of these mutations are independent of the normal surveillance system, mismatch repair system, independent of the so-called error-prone translesion polymerases, dependent on one or the other of these two polymerases, the major replication polymerase. And about 40% of these events have a signature. And the signature is that the polymerase slips or falls off or doesn't know where to go next, as I will show you. Okay, so how did we do this? We tricked up the system by getting rid of the normal mating type sequences here, and we put a functional copy of the uracil 3 gene in this location. But it's silenced, because I told you that this region is silenced. So the cells are ura-. If we turn on the HO, then this is used as a donor, and we put the uracil 3 locus here, and the cells are ura+, because this region is not silenced. And then we could look for mutants. And mutants are easy because they're resistant to a drug called 5-fluoroerotic acid. So we could look for things that were ura-, and we said, so those must be mutations that arose during the switching process. And then reviewer number 3 came along and said, yeah, but how do you know that there weren't mutations already here? So we unsilenced the locus, which turns out to be really easy because this very small molecule, nicotinamide, inhibits the SIR2 histone deacetylase. That unsilences this locus. Now these cells become ura+ again. So that tells us for sure that the mutations arose during the switching process. That was cool. Okay, so we looked at a lot of them. The things above the line are all base pair substitutions. The ones in black are chain terminating. They're nonsense mutations. But everything below the line is some kind of alteration of the sequence. And what we found was that the most frequent alteration of the sequence was a -1 frame shift, in which 3 Gs becomes 2 Gs, 4 Cs become 3 Cs. They're almost all, and they're all -1s, and we still don't understand why there are no +1s. Okay, that's interesting. We actually made a mutation in one of the 2 DNA polymerases, Paul epsilon, and we for the first time saw +1s, but we don't understand why there are no +1s here. They're all -1 slips. Okay, there are some other slips. These ATC becomes 1 ATC, and here is a 65 base pair deletion, which is flanked by 7 bases of identical sequence. And somebody named George Streisinger a long time ago suggested that these were events where the polymerase jumped from one site to another and made some either duplication or deletion. So we could see all of those events. So those were interesting. And by the way, this is exactly the same mistakes we make. Okay, so there's a word for this in the manuscript business, where you should be copying 3 copies of a word and you end up copying 2. That's what the polymerase is doing. Okay, that's how you'll remember this, right? Okay, but okay.
How often does this occur?
So this is 1,000 times higher than the baseline, but it's still only happening 1 in 10 to the 5th cells. Okay, but to put it on your evolutionary scale, that's a big increase. Okay, so then we found events that at the beginning we just technically called weird and totally weird. The weird events do multiple things, change base pairs and also delete or add bases like these. And we worked out that virtually all of these events that are in the little blue boxes could be explained by something called quasi-palindrome mutagenesis, which is an event I still don't believe happens. Okay, because it's just too hard to understand. So what it means is that here's the polymerase going along, copying the donor. This whole stretch of DNA has to fall off. And then it can self-complementary base pair, making what is called a partial palindrome, a quasi-palindrome. And it starts to copy itself. But the dynamics of the system is going to force it eventually back into being not a cruciform. And so it goes back to copying what it ought to be copying. Okay, so that means that this whole piece of newly synthesized DNA had to fall off, do this thing, and then re-complementary base pair in order to make this happen. And these events happen a lot. Okay, they're not the majority of the events, but they're real events. Okay, and so when we published this at the same time, some paper that et al., Garber et al., a huge number of authors, published the mutational processes of these breast cancers, and what they said was they found events which had complex mutations at the sites of chromosome rearrangements, and they thought we were actually providing them with some kind of a justification for these events. So I'm not sure that's true, but we in our grant applications, we say that. Okay, so the very weird events were completely different. They had DNA sequences that didn't come from the template. Okay, so what were they? And so this is a lesson for students about being lazy and lucky. Okay, lazy was that we didn't put the Saccharomyces uracil 3 gene here. We borrowed one from Jasper Rhine that was Clavieromyces lactis, and it's only 72% identical to the uracil 3 gene. And we knew that the yeast cell had a uracil 3 gene, but it was interrupted by a transposon, and it's only 72% identical. So we said, what could possibly go wrong? Okay, so that's the lazy and the first aspect of lucky. Okay, so because we didn't remove these sequences, we found these events. Because what is happening again is that the DNA being copied must fall off, go to a completely different chromosome, find matches, and start copying these sequences, and then it has to come back. It requires 2 jumps in order to eventually be recovered in in our system. And so that was extraordinary, but it's made even more extraordinary by the fact that if it just went here and copied and went back, it would be Euro plus, because there's no discontinuities in the two 72% identical open reading frames. And so it turned out that every event that we found used the same 5 base pairs, either as the beginning or end of the jump. And that happens just by chance to be 3 codons that happen to be arranged in such a way that there are 5 base pairs of frame-shifted sequence. So how many? That's 1 lazy, 2 luckies, or I don't know how many. Anyway, so it's just amazing that we found these events. Okay, so once we knew we could find them, postdoc Olga Zabanina came to the lab. My granddaughter drew this picture for template jumping from one place to another. And the idea now is we want to study these events and only these events. And so how do we do that? We put a 32 base pair deletion in this place. And now when it jumps over here, it's still going to be Euro minus. But if it goes here to here to here, it makes a chimeric gene, and now it can be Euro plus. We know how to select for Euro plus. Okay, so Olga selects for Euro plus. And these events, again, are not that frequent, but they are easily recovered. And interestingly, reviewer number 2 had said, maybe the reason you're getting these events in the first place is that you're forcing the polymerase to go through these highly ordered nucleosomes, and that's why it's falling off all the time. But now we can unsilence this locus, and it's still Euro minus. And so we did that experiment. We unsilenced the locus, and the rate doesn't change. So the instability of the polymerase going down the DNA is not because it's having to traverse a highly ordered set of nucleosomes. So we've measured how often they happen. The amount of DNA that they synthesize in these events are variable, roughly 250 base pairs on average, but there's no beginning or end place for these events because we don't have the 5 base pair restriction anymore. And so there are lots of places where the event can start. If we look at one place where the event starts, there are many places where it can finish. So there's no preferential beginning or end. It's not quite true because some sequences are used very frequently. And they are most frequently sequences that have the most homology. These are all microhomologies that are serving as the anchor points for these jumps in or out of the sequence. But here's a sequence of equivalent length, which is incredibly rarely used. And we do not yet know what exactly are the rules to say which sequences are being used frequently and which are not. This is a bioinformatic problem I would like to pursue more. Okay, and the other thing that Olga found was that this is what happens when they're 72% identical. What happens if you make them fully identical? So all we do here is replace these sequences with a promoterless copy of the same sequences. There's now a 10,000-fold increase in these events. They are now happening 3 in 1,000. Okay, so now we're talking real numbers. Okay, and that says that what we can infer from that is that the polymerase is falling off all the time and getting back on probably most of the time accurately, but it has this tendency to fall off a lot. And so I think that's kind of amazing. Okay, so at the same time, Dujon comes along, and they are looking at other kinds of jumps and rearrangements, which are somewhat similar to what I just told you. And so what Bernard's lab started with, Romain Kozul started this work, and then Celia Payen did a lot of work on it, is that there are 2 copies of the ribosomal protein gene, RPL20, and if you delete RPL20A, the cells have to get by with only 1 copy of this ribosomal protein gene, which it really hates, and they grow slowly. And so the cell finds one way or another to duplicate this sequence so it has 2 copies. And so many of these turned out to be intrachromosomal tandem repeats. Others of them turned out to be interchromosomal hybrid chromosomes that had duplicated these sequences. And very often, there are repeated sequences at these junctions, as illustrated here, that allow these events to happen. Is that a fair summary? Okay. Okay. But some of these events are also microhomology mediated of the sort that I showed you. And so they have relatively small numbers of base pairs at their junctions. And they turned out to be mechanistically identical to events that were described in human cells at the same time by Jim Lupsky's lab, which they called microhomology mediated break-induced replication. And Bernard's lab called this MMIR rather than MMBIR, but we know them to be the same. Okay. And so we wanted to study these events. And so another postdoc, Ranjit Anand, decided we could study them. We want to find cases where we can jump from one template to another. And so here, what we're going to do is break-induced replication. We make a break. The end of this break finds homology to another piece over here, 300 base pairs matched. And then it copies, and then it has to jump again and find 300 more base pairs which it matches, and go to the end of the chromosome, and voila, it's Euro plus. We know how to score Euro plus. So this is really easy to score. And it turns out these events are incredibly frequent. Again, because the polymerase is unstable and is able to jump and somehow find homology elsewhere. It finds it better intrachromosomally than interchromosomally, but we don't have enough data about the interchromosomal ones to know what the real rule is yet. So we can do this with sequences that are either perfectly matched or with sequences that are highly mismatched. And when they are highly mismatched, we again see these microhomologies being used as the place where things start. I'm not going to tell you all the crazy things we found, except to say that the first jump has rules that say use this sequence most of the time that the second jump doesn't have. The two jumps are not, one is an invasion and a synthesis, and the second is a jump. They're not the same event. And that's all I'm going to say about the difference. But the last thing that Ranjit did was just putting half of the uracil gene here and the rest of it here. If we turn this on, it ought to copy this way. And maybe it would keep going around and around and around. And Bernard is going to tell you that that's something that happens. But we said this should be lethal because it's not going to get the rest of the pieces of the chromosome that it ought to have. But the answer is it's not lethal. And it's not lethal because as this copying goes along, it finds repeated sequences. And when it finds those repeated sequences, it can jump from that repeated sequence to another chromosome and copy from there all the way to the end. Okay, so these are translocations from one chromosome to another that will cause the chromosome to be stable and to pick up all the missing pieces that it needs for this to happen, which we did with Tom Pettis and his technician, Patricia Greenwell. The most interesting of these events is this one. Okay, and this is, again, the break is here. It starts copying this way. It copies through some repeated sequences, which in yeast are called TY elements. But there's another part of a TY in the other direction, and it turns around and goes back through the same sequences. And you end up with something that Jim Lupsky calls complex triplication rearrangements mediated by inverted repeats in the human genome. So we have exactly the same events in yeast. And what you're going to hear about from Bernard is that there are very, very complicated events that I think are somewhat similar to this, if not identical, which his lab has described and which has been recently published. So congratulations to Bernard. This wonderful piece of artwork involves 3 different strains of Candida. But 3 pathogens, yes, which is just, I think, an amazing picture, which you can copy and show your friends. Buy my book, yeah, well. Okay. So, in answer to your question, that's the book.
Pleasantly surprised.
Yes, well, yeah, this is an actual quote from Steve West, yes. And I mentioned the guys in my lab doing the work. It's just a great pleasure. I've known Bernard now since the early 90s, and he he continues to be an astonishing scientist. So thank you very much. Yes, Kirk.
So, two questions. One was whether the direction of the replication fork that's coming into the genes makes a difference.
We don't know that. We haven't faced up to that question.
And then the second, maybe simpler question is, so we've got this three-dimensional map of the nucleus. Is there, and you made all those mutations, or is there any confirmation of that map in your strains using like these black operator arrays?
No. We took the map, which was done on a somewhat different strain. It's quite related actually to S288C, what we work on, but we took that map for what it's worth. Now in mammalian cells, when you put the cells in G2, where the chromosomes condense in metaphase, all these interchromosomal contacts are almost all erased. That's Job Decker's work. We have done this experiment for G2 arrested cells in Cerevisiae, and the bad donors are still bad and the good donors are still good. So we didn't do many of them, but we did some. And we think, which is not surprising, Cerevisiae chromosomes condense just a little bit, and apparently not enough to destroy the interchromosomal contacts. Because the map was made from cycling cells. Yes?
So with the polymerase falling off at a rate that you consider pretty frequent, do you think that represents physical constraints, or do you think that's optimized based on selection? And that leads to a related question, which is, have you considered trying to mutate the polymerase to optimize the rate of fall?
Okay, well I didn't put the slide in here. So we actually we have mutated the polymerase delta. And let me, it'll take too long. Okay, we knocked out the proofreading activity of polymerase delta. And we thought, well, now we'll have even more unstable events. And it was exactly the opposite. We have many more base pair substitutions, but all those things, quote, below the line go away. Okay, so now if you look at some biochemistry done by Peter Berger's lab, the experiment is the following. You have a piece of DNA with an oligos at one end just sitting there, and another oligo at the other end, which is the primer. And the polymerase gets on and it copies. It gets to the second blocking oligonucleotide, stops dead. Polymerase delta by itself cannot displace this oligo. The proofreading defective one races all the way through to the end. And the idea is every step in replication is really two steps. You have to synthesize, check the base, synthesize, check the base. The proofreading guy doesn't do the check and apparently is more processive. So we actually, by accident, created an error-free polymerase by killing its error-prone domain, okay? So we think that, in fact, it really is a question of processivity, and that the intrinsic instability comes from the fact that the polymerase has this kind of mode. The other thing is all the events I talked about are pCNA dependent. pCNA is the clamp. And all I can say is the clamp isn't a very good clamp unless it's in the context of a real replication fork. But when it's in this one strand at a time synthesis, the clamp just doesn't hold anybody to its task. Yes?
Where's red 51?
So RAD51 is the guy that sort of initiates the search for homology, allowing the new DNA synthesis to start. We are not yet convinced whether RAD51 is necessary for the jumps. We would think so, but kinetically not. In other words, we know how long it takes to do the search. And these jumps do not take an additional period of time as long as the initial event. So once it started, somehow these new jumps happen quickly. That argues against the idea that RAD51 is doing the same thing. But we do have some data that says that RAD51 is important, but less so than for the first event. And that's a big mystery. Is it the polymerase itself that is just establishing where it ought to go, or is it another recombination mechanism. So we have one crazy mutant in a gene called RDH54, which Valentin will know is a key recombination protein in meiosis, where it works with RAD51's cousin called DMC1. But in mitotic cells, RDH54 has a very minor role. RAD54, its cousin, is essential. Can't do any recombination without RAD54. RDH54 has no problem doing a standard gene conversion event, can't do the jumps. Okay, so somehow RDH54 is defining for us a jump-specific complex about which we know very little.
I seem to recall seeing, and correct me if I'm wrong, but there's some reports that there's mitochondrial DNA or homology segments of mitochondrial DNA is often found in patches.
In the yeast nucleus?
Well, and also during repair that is sometimes used. And there are lots of examples of this now in human genome where there's enough homology to be more than random. And I'm just curious whether what you think of that and whether you're thinking
we are trying to find super rare events of that sort. If the rule simply was you jump somewhere, find 5 base pairs, copy 300 base pairs, find another 5 base pairs, and go back, there ought to be 1,000 of these events in the yeast genome. There aren't. So we're forcing the system to look for what events other than the ones we have defined are there. We have not found mitochondrial DNA. Maria Riquetti, who's in Bernard's lab, did a tremendous amount of work to show how mitochondrial DNA could be captured at the sites of double-strand breaks or inferred double-strand breaks. And we also had shown that we could find evidence of mitochondrial DNA. And also the event that we found surprising was we got very frequent, these are now non-homologous end joining events, but we found 95 base pair pieces of the transposable element. They're the strong stop. They're the first tRNA-mediated piece which apparently floats around and can be grabbed. So the end joining mechanisms, as I said at the very beginning, are completely promiscuous in a way that the homologous recombination mechanisms are not. Yeah.
So did you go back to the map and look whether the homologous donor sequences are nearby in general, or do they can't probably?
In this particular case, they're kind of neutral. They're neither super good or super bad. And we've thought of doing some of these things again in a different context, just to explore that question, but we haven't got that far. Yes?
So it looks like there was a decrease of repair when that donor sequence was near a centromere.
Yeah.
So you're thinking that that's because they're sequestered away.
Yeah.
Is it just that they're away, or is there some active mechanism to prevent the recombination?
No, I think it's just that they're away, because some experiments I didn't mention that were done by Emmanuel Fabre, who's also from Bernard's lab, and Martin Kubiak showed that if you had 2 sequences relatively close to their centromeres, their recombination rates are not so bad. Okay, but getting from there to somewhere else in the genome is apparently a problem, especially intrachromosomally, and which is the place we've really measured it. Yes.
I'd like to know what's going on about chromatin and those proteins that on DNA and the sort of accuracy or the frequency or size of recombination.
You got an hour? It's a big question. As I said, the one thing I can say definitively about the chromatin is that it is not a barrier to using a sequence as a donor, and it is not the cause of these dissociations, because we have these highly organized nucleosomes which are not the reason why it's falling off all the time. We have other experiments which I didn't talk about, where we want to know how the chromatin is reestablished at the site of the break. That's really cool, because a break is made, the nucleases chew, and the nucleosomes go away. Now you repair it, the nucleosomes have to come back. And so we've measured the kinetics of that. We've identified some chaperones which are critical for that, and we're sort of in the process of trying to really understand what chromatin modifications do you need to fully establish the chromatin back at the original site. So we're working on those kinds of problems, but I chose not to add it to this.
Why don't we take a break, otherwise we're going to run out of time here. This year will be available during the break.